All right, so let's talk about the actual lymphocytes, the ones that are involved in, excuse me, adaptive immunity, also known as specific immunity. We have the B cells, we have the helper T cells, and we have the cytotoxic T cells. Let me get yellow again. All right, something that you have to understand, each one of these cells has a specific job. B cells have a specific job, cytotoxic T cells have a specific job, but everything in adaptive immunity hinges on this helper T cell. This is the guy that gets everything started. Regardless of how the immune system is responding, this is the guy that has to start everything. Without this guy, without my helper T cell, nothing else works. Now let me explain why that is so important. HIV, human immunodeficiency virus. Immunodeficiency, your immune system is deficient, it doesn't work. HIV infects these cells. Now remember what I just said. Your entire immune system hinges on these guys working, on the helper T cells working. If I'm killing them with the HIV virus, I'm literally preventing my immune system from working. Okay, just to kind of give you an idea. So, in a B cell, on the surface, you have a membrane-bound antibody. But when we're talking about the T cells, you actually have two things. You have a CD4 receptor and a T cell receptor for the helper T cells. CD4 is important. And as a matter of fact, if you've ever known somebody who has HIV, they talk about the CD4 count. It's these cells they're talking about. The cytotoxic T cell, okay, let's rewind. CD4 is basically the bracket that binds to MHC-II. That's why the helper T cells are recognizing and able to react to MHC-II displaying something on its surface. So MHC-II helps to anchor the helper T cell to its target antigenic determinant. Remember, I only recognize one thing. Whatever this variable region recognizes is what I recognize, okay? So remember that for my helper T cell, it's the antigen presenting cells that are displaying MHC-II. It's the person holding the wanted poster. I'm not killing them, they're just showing me something, okay? I hope that makes sense. Now, the cytotoxic T cell, its little bracket here is actually called CD8. CD8, CD8, let me just leave it at that for right now. They also have T cell receptors that have a constant region at the bottom and the variable region at the top. Now remember, CD4 bound and recognized MHC-II. Hello. CD8 is what recognizes MHC-I. Remember, MHC-I is the protein that displays the funky proteins when I've been invaded. So CD8 anchors my cytotoxic cell death T cell to MHC-I, basically allowing my cytotoxic T cell to make sure I'm bound to the thing that I recognize, and it stays long enough, that it actually lets me, as a cell death cell, kill the cell that I'm bound to because I recognize that there is something seriously wrong with it. Okay, so we are going to talk about, in this particular lecture, the helper T cell. We're going to focus on the helper T cell because, like I said, everything else hinges on the fact that the helper T cell gets things started. So what I'm talking about in this thing is right, whoops, went too far, is right here. This is what we're about to talk about. We're about to talk about the adaptive immune response and how the helper T cell is a part of it. And notice in my little family tree of information, or concept map if you want to get all rubric-y about it, helper T cell is the first thing in line. Then it's the cell-mediated immunity and the antibody-mediated immunity. This gets this and this to work. Without this, nothing else works. Okay? And I know I've said that like 75,000 times, but just so you know. All right. The helper T cell can also be abbreviated as CD4 cell or T sub H for helper. The helper T cell is initially created in the bone marrow, and pretty much all I'm doing is I'm shuffling my DNA sequence around, coming up with different ends to put as variable regions on my T cell receptor. It's completely random. I'm not making something specific to something that I've seen. I'm just making random cells, okay? Now, when I make this random selection of cells, they still have to go through positive selection. Do they recognize an antigen? If they do, they pass on to the second part of the test, which is negative selection. I've got to make sure that they don't recognize my own antigens, the self-antigens, or they'll hurt me. So every single one has a unique variable region. So I've got a whole bunch of different helper T cells that go into the body once they do the positive and negative selection and float around looking for their match. Now, we have tons of helper T cells that never see anything like what's on the end of their variable region, and they never ever activate and they never do anything. But it's the ones that do that are important because they're the ones that start our immune response. So pretty much once I pass my two tests, positive and negative selection, I go and I float around the body. Now, let's say you get the rhinovirus, cold, and an antigen presenting cell ingests it and it takes it apart and it starts displaying the parts and pieces on the MHC-II on its surface. Remember, MHC-II, antigen presenting cell, I'm showing you a wanted poster with the description. So I'm displaying the antigenic determinant for the virus. The correct T cell, I should say helper T cell, okay, T sub H, the helper T cell, can come along, the one displaying the variable region that matches the displayed antigenic determinant. Okay, so here I've got my helper T cell. There is my CD4 binding to my MHC-II on my antigen presenting cell. And you'll notice the variable region is for the red leg that is on my antigen presenting cell. So I am specific in what I am doing. So CD4, this little bracket, anchors the helper T cell to the antigen presenting cell long enough so that the helper T cell can basically confirm it's supposed to be activated. I don't want a helper T cell willy-nilly getting activated when it's not supposed to be. So kind of giving it this anchoring point and letting it sit there for a second gives it, okay, yes, we are supposed to be activated. We are supposed to be doing this. So the next steps. It actually involves the antigen presenting cell, this guy, the APC, releasing interleukin 1, which is a chemical signal. So it makes interleukin 1, okay? And it releases it across to my helper T cell. Okay? Let me get all my writing off. So interleukin-1 gets released. It goes across to my helper T cell that is partially activated. Okay? And that helper T cell starts, hang on, sorry about that. Okay, so the helper T cell will start to mass produce interleukin-2 channels. Now, mind you, I released interleukin-1, but now I'm starting to put channels inside of my membrane for interleukin-2, okay? Then I'm going to mass produce and release interleukin-2. Literally, the cell that's making the receptors is also making the thing that's going to bind to the receptors, interleukin-2. Now, this is self-stimulation. The helper T cell is actually going to begin cloning itself because interleukin-2 channels got made and then got bound to. Now, let me explain something. This set of steps has several fail-safes. If I can't bind to this and stay bound to it, I'm not the right helper T cell. If this cell doesn't release interleukin-1, I'm not going to make the interleukin-2 channels. So there's kind of this partial activation and then there's complete activation. Once I get to the point that I've made my interleukin-2 channels and I've started mass producing interleukin inside my cell, and then I release it and it actually comes back and binds. So let me go back. Watch this. It's going to come over here and bind. It comes back and it binds to that channel. Okay? This is the stimulation to start cloning. Remember, I had one of these cells. If you have an invading force in your cell, you're going to have millions, if not billions of them. One cell, one soldier against a 1,000,000-person army is useless. I've got to start making more. So that's what this cloning is about. I need to start cloning. I need to start cloning this cell because this cell is the one that is supposed to be activated. So this right here, this right here is the jump start. You need to start cloning. And I end up with two sets of cells. I end up with memory helper T cells and I end up with effector helper T cells. So let's talk about those two populations. Memory helper T cells. They're put away. Pretty much, let's say that I make, I'll say I make 100,000 cells in grand total. 100,000 cells. 99,000 will become effector helper T cells and 1,000 will get put away till later, okay? So the memory helper T cells are the ones that get put away until a secondary infection. Now remember something, when I started, I had one of these cells that recognized the red leg of this virus. One. And I've made 100,000, 99,000 are going to go and fight for me, and I have 1,000 that are just going to go into hibernation. Once I'm over this illness, I'm going to have these 1,000 floating around the body, looking to see if they see the red leg again, okay? What this means is we have extra copies that recognize this particular target. If we get the same infection, we actually have more seeking helper T cells looking for red leg to seek out and become that first helper T cell to get warned. So instead of activating one and that one having to make 100,000, let's say next time I get this, I activate 10. Okay? Let's just say I activate 10. That means in the secondary infection, if I activate 10, I'm going to be making 1,000,000 effector helper T cells, and I'm going to be making 10,000 of my memory cells. Let's go even further. Let's say I get infected a third time. Well, I already have 10,000 of these memory cells that have been waiting to see red leg again. Let's say instead of activating 10, in that case, I activate 100. Now, that's 100 that are going to make 100,000. That's 1,000,000,000, if I'm not mistaken. So that's 999,000,000 effector cells, and then 1,000,000 memory cells. Do you see how it's building with every infection? The more I start with, the more I have for the next infection to come along. And the quicker my response can be, because I don't have to go looking around the body as an antigen presenting cell for the one cell that recognizes red leg. I literally have 10,000 floating around. I literally have 1,000,000 floating around. I have a whole ton of them that I can just go, here, okay, you too, here, okay, you too. And every time one of those gets activated, I'm making another set of this 100,000. So memory, remember I said adaptive immune response has memory. This is the memory I'm talking about. Now let's talk about the effector cells. I said everything hinges on these helper T cells, right? Well, the effector T cells have two jobs. I need to activate the B cells that make antibodies that recognize what I recognize, which in this case is the red leg. And I need to go find cytotoxic T cells that are partially activated and activate them the rest of the way. So at this point, these cells, this 99,000 cells, begin a massive search for the B cell that will match the invading pathogen. This special B cell can do something no other B cell can. It makes the antibody that binds to red leg. No other B cell does that. So we need to find the one cell and get it to start doing pretty much the same thing. I need it to build an army so that it can start making antibodies. So another job, like I said, this is what everything else hinges on. I have to find the cytotoxic T cells that are partially activated and release interleukin-2 to cause them to start cloning. Remember that over here, this cloning didn't happen until interleukin-2 bound here. We're doing the same thing until this guy comes around and activates that T cell by releasing interleukin-2. Those cytotoxic T cells are only partially activated. That's like putting the key to the ignition in the car but not turning it and turning on the motor. The T cell, the cytotoxic T cell that has the key in the ignition but hasn't turned it over, needs this guy to turn the key so that he can start making clones of himself and build an army that can actually go and kill the infected cells. Okay, so that is here. Helper T cells, they're the instigators or the switch for both the B and the T cells to begin specific immune response. MHC-II foreign antigen complexes on the antigen presenting cell are what they bind to. Remember, MHC-II is specific for helper T cells. So the activation, it uses CD4 to bind to MHC-II and the antigen presenting cell. If everything is correct, the antigen presenting cell releases interleukin-1, which induces the helper T cell to produce interleukin-2 receptors and insert them into their membrane. And then once it does that, it begins cloning. Two populations are created, the effector and the memory cell. The memory cell I always call the sleeper, regardless of which memory cell it is, because it hibernates. It's waiting for the next infection to happen, but now instead of having that one single copy that had the variable region I wanted, I have multiple copies available. The effector helper T cell, I call this the searcher. And I call it the searcher because it goes looking for the B cell that can create antibodies against the invader that activated them. In the case of our example, it looks for a B cell that can make antibodies against red leg. Okay? It also activates the cytotoxic T cell by releasing interleukin-2, which signals my cytotoxic T cell to begin cloning. That just says cling, I mean cloning. Okay, so that is the helper T cell. That's what we've just finished discussing.